have a clicker? Yeah. Perfect. Okay. Yeah, thanks the organizers for inviting me to this gorgeous conference here. I heard this great talk of half an hour ago, and was talking, or we were discussing after that a lot about engineering or re-engineering of the human body, the exoskeleton, for example. And I want to go a little bit into the body and talk about implants, so re-engineering of tissues and organs. And I want to give you one technology as an example with which we try to re-engineer the body. So let's take a closer look first into a tissue or an organ. I have today the heart valve as an example. This accounts also for other organs, but we need to have one example. So this is a human heart valve. It's a section, so a cross-section of a heart valve, and we stained it with different colors. What you see here is that it's a really complex structure, so it's not only a flat membrane, and those little black dotties you see in there, these are human cells, heart valve cells in this case, and these cells, they form their own house, so their own matrix, and they build the ECM, and they are tissue specific. And in some tissues or organs, you have more than one cell type. Also in the heart valve, here you have 2 cell types. And there is the extracellular matrix. Does it work? No? Now it's there. Perfect. So the extracellular matrix. The extracellular matrix is like a house for cells. It is produced and constantly remodeled by cells, and it varies from tissue to tissue. So here you see a lot of yellow and also a few black fibers, and this is different in every organ. And the extracellular matrix is responsible for the tissue strength and also for the durability, so for tissue mechanics. So it's highly important we do not only have cells, we also have a matrix around. And then I call the next point the microenvironment. Depending on the organ or tissue, you have different mechanical forces acting, like a constant blood flow, for example, in the heart valve, where the cells are sheared all the time. We also have, by the movement, we have a strain on the organ. For example, in a bone, you have all the time compression on that bone. So there are a lot of mechanical forces acting on these tissues. So when we want to re-engineer an organ or a tissue, we have to consider that it's not a simple structure. It is highly complex. Let's take again the heart valve as an example. Here we see the aortic valve. It has 3 leaflets, so 3 little parts, and you see it here in the open and closed condition. And when you cut out one piece, you have a picture here where you see how it looks with this radial structure, and it's a thin membrane. And when you then again cut it and stain it, this is the image on the lower end, you again have then this really highly complex structure. And you know what we are implanting at the moment? This is what we place into the human body. And this is actually the gold standard. I mean, there are other options like donor organs, but as we heard before, in a few years, we only have to take taxes. Where do we get those organs from, those donor organs? So there's a lack or a limitation of donor organs, and so we have to use artificial valves. And this is a heart valve which is implanted, and as you see, this has nothing to do with a real valve, with a real environment. And this not only accounts for the valve, this is for all tissues and organs what we implant when we look at hip implants, for example. It's just a stainless steel ball what we place into the hip. So it is tolerated by the body. We are happy that we do not have an immune response or capsule formation. It is tolerated, accepted, but it doesn't really integrate into the human body. So we do not have a tissue growth or remodeling or regeneration. So this valve is a valve. It opens and closes with limitations, but that's it. No regeneration as soon as you need a new heart valve. For breast implants, for example, we know the issue of capsule formation. So the body says, well, the silicone, I don't really like it, but I accept it, so I form a capsule around it, and it's a hard and stiff structure, and you have to do resurgeries and replace the implant. So, and then there are also tissue-specific complications, like, for example, in the valve, due to the disrupted blood flow, what we have in there, we get blood coagulation, thrombus formation, and it can also, with those biological valves which are available at the moment, there's the problem with calcification, so the valve starts to turn into a bone. The question is, why is it turning into a bone? There are some signals going on telling the body, make bone out of it. So this is why in my group at the NMI in Reutlingen, from the University of Tübingen, we have the following strategy. So we have the issue, no regeneration, no remodeling, no growth with current implants. So the strategy is, we want to look into nature. We want to look into the matrix, the ECM, the proteins which are in the ECM, but also into the cells. So what do the cells need, and how do they develop in an embryo towards an adult human being? And then we try to get all these properties and then mimic them in a material after that. So we want to mimic the structure or the architecture. We aim to mimic the tissue mechanics. We want to have also biological signals in there, which guide cells that they do what they should do. And so we aim then to end up with functional implants. So implants, which, for example, can also be implanted acellularly because they have specific capture proteins that they can capture from, for example, cells from the bloodstream. So the vision is that we want to have regeneration instead of symptomatic treatment, what we have at the moment. So again, the extracellular matrix, I like it. This is why I want to show it to you again in a schematic. So it's a complex network. You have those collagen fibers. I think this is known to some of you. Hyaluronic acid. It's also used for aging, anti-aging lotions. And this is all in our body. So it's a complex structure, a fibrous structure of proteins. And how can we mimic this fibrous structure? And here I want to introduce a method to you. It's called electrospinning, a method to generate fiber-containing scaffolds from polymer solutions. So here we have a high electrical field. We have a polymer solution in a syringe. We pump it through. We apply a high electrical field. And so a little fiber forms, the fiber spins down to the collectors, the solvent evaporates, and we get a solid fibrous structure, as we see here in this upper image. On the right side is also an image from the electrospinning process. So we get fibrous structures here, and we can spin synthetic polymers, for example, degradable or non-degradable. We can use natural polymers, collagens, proteoglycans, but also proteins, and we make hybrids. So either functionalizing the materials after the generation process or incorporating proteins during the electrospinning process. And then we can use these scaffolds as implants, test system, humanized test systems, or as drug release systems when we encapsulate a drug, for example. For the heart valve, I want to show you a little bit of data. So this is the structure, actually, which we electrospun for the heart valve. So this melted together fiber structure we got via UV cross-linking. And here we nicely see we have this fibrous structure, and we can also modify the surface. The mechanical characteristics are almost the same like in the natural heart valve. So we were able to mimic the strength and also the durability of a heart valve. We placed it then into a bioreactor, and as you see here in this little video, the valve, this is the material, the re-engineered organ, and it opens and closes properly and under physiological conditions. So this means it's the blood pressure of 120 to 80 millimeter mercury. And then in the next step, I functionalized the material, and here this colorful structure, this is a protein, so the upper image is with protein and the lower one without, and in the one with protein, we see that a massive amount of cells attached, so it's one big agglomeration of cells, so the cells got captured from the material, whereas we have almost no cell or only one single lonely cell with the unmodified material. And what we saw nicely was that the matrix which developed on this material was pretty similar to the matrix of an 18-week embryo. So after 6 days in culture, we were able, with the material, with a specific material, to induce an embryonic-like behavior. So I want to show you a few other examples, just that you get an overview of what else is possible, because I'm only talking about the heart valve. We can also use this technique to encapsulate drugs. So in this case, we have, for example, encapsulated diclofenac. I think most of you know that. This is like paracetamol. A lot of people take it for pain relief. We have a release of this protein within 48 hours. It's meant as a patch for the skin, and as soon as it is in contact with the wound, the fibers start to degrade, and the protein is released constantly. Another idea is the immune response. So we work currently in a project where we think about what's the immune response. It's a really big topic, and I don't want to dive too deep into it. It's too complex, but without an immune response, we never get regeneration, so the immune response is important. But we don't want to have a, let's say, bad response. How can we use materials to influence the immune response? How can we functionalize implants, possibly with different fiber sizes, different topographies, in order to get a material which causes a good immune reaction, so a regenerating immune reaction? And then, as I said before, there are the test systems, so we can form elastic fibers with human cells, for example, on a material. So this is thought to mimic aging at one point. Here we can form elastic fibers, and it's called the elastogenesis. But then I'm also working on a heart valve calcification model, so where we have a heart valve in a dish, re-engineered, and with human cells, and where we can induce calcification. And then see how can we treat this calcified valve in order to get repaired. Or another idea is heart muscle regeneration after myocardial infarction. Here we have a system where heart cells, they start beating, they beat properly, they respond to different drugs, and now we're aiming to induce an infarct now in this model in order to see how can drugs act in this system and how can they help to regenerate the heart muscle. And then it's not only the heart valve, there are also other organs. I like the cardiovascular system actually, so I also work on blood vessels, so electrospinning of small vessels, smaller 6 millimeters for bypass surgery, for example, because there is no adequate material at the moment. So we have to take our own veins and implant it from leg or arm and implant it into the heart. So here there isn't even a material, we need to engineer a new one. Myocardial patch, drug or protein loaded, which we can place then on the heart, and a special proteins are released to induce regeneration. We work a little bit on tendon repair or tendon regeneration, and also the electrospun scaffold has a really high potential as a hernia repair material, and this is what I love to look deeper into it. This is my next goal, to look for hernia repair after the heart valve. So I thank you for your attention, and I'm happy to discuss you further.